Good morning. The topic is ground tissue system, or it is called fundamental tissue system. In ground tissue system, there are a number of tissues which are getting differentiated, and usually they are of four types. First one is cortex. Cortex, inner to the cortex. The cortex contain hypodermis, general cortex, endodermis. Then these three are the part of cortex, and the second one is pericycle, and the third one is pith, and fourth one is medullary rings. So this is the part of or components of ground tissue system or fundamental tissue system. Okay, coming to the first one, cortex. I have mentioned that already the cortex contains three different tissues. That is the dedifferentiation of cortical tissue into three major anatomical areas, which is called hypodermis, general cortex, and the endodermis. Okay, first one is cortex. What is cortex? The cortex is the zone spread from the subepidermal layer of plant body, and the layer to the layers enveloping the stele, that is the central cylinder. So which is called the cortex. In a plant body, it is with the two major areas, that is outer cortex and inner or middle stellar portion. So the entire middle stellar portion is covered with this cortex. In dicot stem, cortex is generally differentiated into hypodermis, general cortex, and endodermis. These are the three anatomical differentiation of cortical area in a plant body. Okay, coming to the first derivative of cortex is hypodermis. Hypodermis is present just below the epidermis. In dicot stem, it is collenchymatous, while in monocot stem it is sclerenchymatous. That is the difference between the dedifferentiation of hypodermal tissues. Once again, in dicot stem, usually the hypodermis is collenchymatous, and in monocot stem it is sclerenchymatous. Okay, the collenchymatous cells contain chloroplasts and are photosynthetic in nature. Chloroplasts may not be found in all collenchymatous hypodermis. So, in certain species or certain numbers of plants which show the hypodermis which is pigmented or chlorophyllated. In the green stem of monocots, hypodermis may be chlorenchymatous instead of sclerenchymatous cells. So, this is the nature of anatomical structure of hypodermis. Then coming to the second derivative of cortex is ground cortex. What is ground cortex? This is the region between hypodermis and endodermis. It is usually composed of loosely arranged parenchyma cells, that is the cells with the intercellular spaces. And some of the species which do not show such loosely arranged nature, that is compactly arranged general cortex is also seen in some species, and that means the cells do not show any intercellular spaces. Okay, the cells contain, that is in the general cortex, the parenchyma cells contain starch grains, and different types of crystals are found in these cells. And the sclerids, resin ducts, tannin cells, oil cavities, and latissiferous tissues are also present in the general cortex of the plant body. So, normally in the distribution of cortex, what we can see, the structurally it is parenchymatous, but it occupies such ergastic substances or such type of depositions, which is already mentioned, that the sclerids, resin duct, tannin cells, oil cavities, etc. And even latissiferous cells also present in the general cortex, that is inner to the hypodermis. Okay, in roots, This is such a differentiation of or such deposition is usually seen in stem. But while coming to the root system, the cortex is homogeneous and consists of only parenchymatous cells. So here you can remember that what in plant stem is heterogeneous, that is different types of tissues are differentiated or de-differentiated in the general cortex. While coming to roots, it is only homogeneous, that is only one kind of cells is usually present and they are normally the parenchymatous, involved parenchymatous cells. It's a main function of the general cortex is to store food materials and to give mechanical support to some extent. So this is the second derivative of cortex. And coming to the third derivative, it is called endodermis. It is the innermost layer of cortex, and this layer is made up of compactly arranged parenchyma cells which appear tubular or barrel shaped in cross section. So how we can differentiate these endodermis and the general cortex is simply nothing but the cortex or general cortex contains loosely packed parenchyma, but endodermis is compactly packed tubular or barrel shaped cells. Okay, the characteristic feature of these cells is the presence of bands or strips of suberin on their radial and transverse walls. That is, the vertical walls and inner walls become deposited with the suberin. What is the peculiarity of suberin? Suberin is not soluble in water, and such suberin is deposited on the walls of these endodermal cells, and they are commonly called, because of the suberin deposition on the walls of endodermis, they are commonly called Casparian strips. So what are Casparian strips? It is the suberin deposited thick walls of endodermis, which is the third derivative of cortex. Okay, and what is wherever it is distributed, the endodermis is usually seen in the stem of most herbaceous angiosperms, aquatic plants, repose and rhizomes. Okay, but it is absent in woody stems of dicots and gymnosperms and in leaves of angiosperms. So usually the herbaceous plants of angiosperms showing the clear differentiation of the third derivative of endodermis, but as in such dicot plant with a prominent stem, and even in gymnosperm or leaves of angiosperm, the cortical tissue or the general cortex, inner to the general cortex, the endodermis is usually absent. Okay. Then the second component of ground tissue or fundamental tissue system is pericycle. Coming to the pericycle, pericycle surrounds the vascular tissue where it is present. It is at the central zone. Actually, the cortex is covering or encircling the stellar portion. So, pericycle is the second derivative of say ground tissue system, and usually it surrounds the vascular tissues, that is xylem and the phloem, and it forms the limiting layer of steel. So it is also considered as the outermost layer of the steel. Pericycle is considered as the outermost layer of steel, but endodermis is similarly the innermost layer of the cortex. Okay, it may be single layered or many layered. The pericycle region may be homogeneous or heterogeneous. So such a differentiation we can see in the pericycle development. Homogeneous pericycle consists of parenchyma only, as seen in all roots and pteridophytes. But heterogeneous pericycle consists of patches of parenchyma or sclerenchyma, as seen in dicot stem. That is the structural differentiation of pericycle, what we can see between in roots and the shoot or stem. Okay, in roots, the pericycle give rise to lateral roots. So in roots, where we can see there is a taproot system which is produced generally, but there the plant which produce lateral roots, really it is developed from the pericyclic cells. In stem, during secondary growth, phellogen or cork cambium arise from it. That is, the development of extrastellar thickening in stem. It is enunciated by these pericyclic cells, which forms the phellogen or cork cambium. That is, which ultimately develops to form the periderm formation. Okay, and so that is why it is concerned with the secondary growth in thickness also. So pericycle has a major role or the particular role in the secondary growth and the extracellular thickening in plant body. In certain plants where anomalous secondary growth is found, it forms the secondary cambium. This pericycle is considered as the secondary cambium in those plants showing anomalous secondary growth in thickness. Okay. Then the third derivative of the ground tissue system is pith. What is pith? Pith is the more or less cylindrical ground tissue, that is the parenchymatous tissue, which forms the central core of the stem. That is, inner to the stele, there is the middle portion. What we can see, it is the parenchymatous in nature also. It is externally bounded by vascular bundles. So, inner to the vascular bundles, we can see a ground area, and which is concerned or which is developed by parenchymatous cells, and which is called pith. Okay, it is made up of loosely arranged